Hey you!
What?
Do you have a bio test coming up?
Yeah.
You need to study! You better study up! Here's a brief summary of what you should know. Just kidding! I'll try to highlight the important aspects briefly. Things you should remember about Hershey and Chase is that they use viruses and that the type of transmission was transduction, the transfer of DNA from virus to cell, and the DNA of the viruses caused other bacteria to change. And things you should remember are that a blender was used and that radioactive isotopes were used as well. So Frederick Griffith was another scientist, but he used bacteria in different samples and injected it into mice to show how the DNA from the bacteria causes another bacteria to change. And transformation was used to show the incorporation of the foreign DNA into the mice. In protein synthesis, there must be a triplet code. So in DNA, there are base pairs, adenine and thymine are base pairs, and guanine and cytosine are. But in RNA, it's adenine and uracil are pairs, and still guanine and cytosine. So the sequence of events that happens in protein synthesis are begins in the nucleus, that's where the genetic code is contained, and then the mRNA is where proteins are coded and made inside the nucleus and travels through the cytoplasm. And then it goes back to the ribosomes to build the proteins, and then tRNA transfers the amino acids back to the ribosomes. From the nucleus to the mRNA, transcription occurs, so the original base sequence would code for its pair in the mRNA strands. So, for example, if the first sequence was ATT, in the mRNA strand, the code would be UAA. Now, regarding gene technology, restriction enzymes are the proteins that cut DNA at certain restriction sites. Restriction enzymes are also known as endonucleases, so those words are synonymous. And specific types of enzymes you should be aware of are EcoR1 and HEY2, and that restriction enzymes can fragment pieces of DNA into differently. So the blunt ends are harder to attach because there's nothing exposed, but the sticky ends are easier because their bases are exposed. Specifically for EcoR1, this enzyme cuts immediately after the G in the sequence GAATTC, and it reads simultaneously with the second strand, so it would cut in the same locations. So second strand is CTTAAG. The cut would be between A and G. The end result is recombinant DNA, and the purpose of that is so that people can change the genes of organisms.